Yeah.
Impressive. Impressive. Oh, terrific. See, we've got just another one or two who are coming in from the waiting room. All right. Well, I think we're ready to start. Let me see if I see any other notes. Great. Well, welcome. We are so pleased. I'm so pleased to welcome you here to our kickoff of our Sip of Science here from the Biodesign Institute at Arizona State University. My name is Carrie Robinson. I'm Senior Director and Chief of Staff here at Biodesign, going on my 12th year. So feel pretty passionate about this place and what we do, and couldn't be more grateful than to be here with all of you tonight as we kick off this incredible series. I am going to share my screen here because there are just a few housekeeping things that I want to be able to share with you here tonight. And here are some Zoom functions for tonight's meeting. So in case, I feel like now we're all very familiar with Zoom, but in case you're not, please know that there are ways to display, change your display name. And mute, if you can mute your audio, that would be great. Just want to be sure that you can hear our speaker and everything that we have planned for you tonight. And then also to tell you to this chat function. So if you scroll at the bottom of your screen, you can kind of roll up and you'll see this chat function. If there's a burning question you want to ask, you can just put that in the chat, and those will be teed up to Josh so that he can respond to those. Also, at the very end, we'll have just an open Q&A session. So, you'll be able to ask questions. Also, feel free, if you'd like, to share your video. Part of it is we want to see all your amazing, wonderful faces. So if you feel comfortable sharing your video, we would love to be able to see you. But if you can just mute your audio, that would be terrific. So I want to start things off by just sharing a little bit about this great place that is the Biodesign Institute. It is the premier scientific research institute in the nation's fastest growing research university. At the Biodesign Institute at ASU, we address an expansive array of global challenges by creating nature-inspired solutions, including new vaccine discovery and delivery. You're going to hear more about that. Early detection and treatment of cancer and other infectious diseases. Techniques for detecting and removing contaminants from the air and water, and also the application of nanotechnology to biomedicine and electronics. So today, about 1,200 biologists, physicists, chemists, engineers, mathematicians, and others are blending their knowledge to address society's greatest challenges in health, environmental sustainability, and national security. And that, in a nutshell, is what we do within 17 centers of research at the Biodesign Institute. And you can see our website is here. So there's a whole wealth of information to share with you there. So let me share a little bit about this award-winning series, Sip of Science. So Phoenix Magazine named this one of the best places to eat, learn, and drink in Arizona, in Phoenix. And so we're really excited about that. Now, of course, for many of you who these familiar faces I recognize who have been with us, this is our 4th year, and we would most want to be with you in person, but we've got the next best thing. And that is, we started to reach out to find a local restaurateur or mixologist to come in and help us talk about this topic and prepare something wonderful. We were excited that Hush Public House, they came up with their own version, and you guys have seen this, but their own version of a cocktail just for us tonight at Biodesign, and that is their Quarantini. So we hope that you have your ingredients at the ready. And I'm going to go ahead, and they prepared a little video to share with us. You'll want to maybe turn up your audio a little bit. The audio wasn't as perfect as we had hoped, but they have something pretty neat in store, and I'm excited to share that with you now. Here we go.
How you doing? I'm Charles Barber, co-owner of Hush Public House in Scottsdale. We're located on Scottsdale Road, just north of Thunderbird. Today we're going to have Kayla Reed, our mixologist, making you a quarantini. Really good cocktail. We are open for dinner Tuesday through Saturday from 5 p.m. till 10 p.m. and Sunday for brunch from 10 a.m. to 2 p.m. We really look forward to having you here for curbside, takeout, or social distance dining. Thank you so much.
Hello everybody, I'm going to show you how to make the quarantini today. So you'll take your small shaker, we'll start with the Cointreau here for you, and we'll do 1 ounce. Dump there. Cranberry juice, you'll do 1 ounce. The gin, 1.5. Juice, you'll do 1/2 ounce. Then you're going to put a scoop of ice into your shaker. Then you'll shake it for 10 to 15 seconds or until this gets nice and icy. Dump it right into your lovely martini glass. Perfect. And the best part, top it with a little bit of champagne. And you're going to garnish it with an apple slice. Floats right on top, your lovely quarantini. You can also make this fun drink a mocktail if you wanted to. You could omit the gin and omit the champagne, and instead of doing the champagne, you can do an orange flavored soda water like LaCroix or any other brand that you can find and make it a mocktail.
So a huge thank you to Hush Public House. And as I was learning a little bit about Hush, I didn't realize that they not only is it upscale kind of intimate dining in North Scottsdale with their restaurateur and chef, Dom Ruggiero and Charles Barber, but they have been getting rave reviews for one of the best restaurants in North Scottsdale. And Phoenix New Times and others, they were voted as best new restaurant in 2019 and all kinds of things. So I'm wondering who made a drink. Mine's a mocktail, but cheers.
I see that Tim Long did the real deal. He got it.
I missed it.
I saw him shaking the ice there.
Nice,
Yeah.
nice. Well, everybody,
Mine a little earlier.
here from the Biodesign Institute.
Cheers.
Awesome. Well, many of you know our incredible distinguished executive director, Dr. Josh LaBaer, and I feel really honored to have been able to work with him for many, many years now. For those of you who might be new to us in this series, I just want to provide a brief bio, and then Josh, we're going to have you take it away. So, as many of you probably know, Josh is one of the nation's foremost investigators in the rapidly expanding field of personalized diagnostics. Now, he was recruited to the Biodesign Institute at ASU and was the first Piper Chair in Personalized Diagnostics in 2009. And previously, he was founder and director of the Harvard Institute of Proteomics. Now, we're so pleased to have John McGarity on with us here tonight and thank him for his distinguished career. And Josh was recently honored for bringing teams together to address some of the world's greatest challenge, and he actually won the John W. McGarity Arizona Bioscience Leader of the Year Award by AZ Bio just recently. And I see that Joan Walker is here as well, so appreciate that incredible support. The last little bit to share is that under his leadership, the Biodesign Institute won Innovator of the Year for Academia at the Arizona Governor's Celebration of Innovation Awards just recently. There's a whole laundry list I could share about Josh, not to mention that he's just an incredible, wonderful, and brilliant human being. But we're really honored that he's able to kick off this series tonight. And with that, he's going to share with you Innovating in the Midst of a Pandemic, Time for a Spit Take. Josh.
All right, let's get this, see if I can get this going here. All right. See if I can. Okay, so those of you who are familiar with our format here, I blew that, didn't I? All right, let me go back to this. All right, know that this is really meant to be informal, and hopefully, you'll be able to ask questions during the talk. Feel free to type them into the chat, and then as soon as the talk is over, we'll, go back to the Hollywood Squares thing, and we'll try to talk more personally, so that we don't get encumbered by the slides. So I want to talk to you a little bit about what we've been up to this last year. It has been a year like none in my lifetime, that's for sure. This has been quite a challenging year, and the pandemic, the COVID-19 pandemic, we've not seen anything like this on planet Earth for over 100 years. So it's been a shocking period for all of us. The virus, the SARS-CoV-2 virus that causes this pandemic, I like to say that it won the trifecta in terms of being a nasty virus. And by that, I mean, it sort of has the 3 S's of severity, spreads fast, and stealthy. The severity took us by surprise. And, initial reports, many of you may remember about a year ago right now, we were hearing that it was a little bit less than the flu, but in fact, it's much worse than the flu. It's at least 10 times worse than the flu. It's a challenging virus because it has such a disparity in the way people experience it. On one end of the spectrum, it is the leading cause of death in Arizona now. It is a huge killer. It takes lives like crazy. But on the other end of the spectrum, some people don't even feel it at all. And I think that makes it a particularly challenging virus because some people who get it don't even know they have it. And those people can carry it and deliver it to other people. And that makes it a very challenging virus to look at. It spreads extremely fast. That's the second characteristic that makes it so bad. It spreads in droplets that come pretty much from when we speak, when we breathe. When human beings breathe and talk, they produce 2,600 droplets per second from their mouths of moisture that comes out. And the virus surfs along those droplets of moisture and travels from one mouth to the next person who breathes that air in, and then it gets into the lungs of the next person. So the fact that this travels by an airborne route and it travels in these tiny droplets that can linger in the air for minutes makes it a particularly easy virus to transmit. And I guess the third characteristic that makes it so challenging is that it's stealthy. The peak of infectivity for this virus is about 2 days prior to the onset of symptoms, if you get symptoms at all. And that means that people who are spreading it don't know they're spreading it. The related viruses to this virus, the 2 closest relatives, are the SARS virus, the original SARS virus, and the MERS virus, the Middle Eastern Respiratory Syndrome virus. And both of those viruses are also deadly, as this one is, but both of them have taken much fewer lives. And that's because when people are infectious with those viruses, they're very ill. They feel sick. And so it's easy to identify who is spreading the virus in the case of those 2 viruses. But in this virus, we can't tell because people are spreading it before they know they have it, or perhaps they never know they have it. So the transmission of this virus, we say, is exponential. What that means is that each person who gets it can transmit it to up to several people. It looks a little bit like this. Imagine if one person has it, that person could give it to somebody else. Meanwhile, that person could give it to somebody else while the first person could give it to somebody else. And then so on and so forth, each person transmitting it to other people. What we at Biodesign realized early on, and that this is really just about a year ago now, was that if we could interrupt transmission, we could save lives. If we can get it back to this point here and prevent all these additional branches on these trees, we could prevent a lot of people from getting ill and dying from the virus. And because we knew that the virus was transmitting before people knew they were sick, the only way to identify if somebody has this virus is to test for it. And we knew from early on that testing was key. And I had a conversation just a little bit less than a year ago with President Crow at the university. And he was asking me, he said, look, we have 100,000 people in the ASU community. How are we going to test all these people? And it was at that time that we realized that the technology that was used to test for this virus is something that my laboratory actually had in it. We had used it for developing a test for radiation exposure. We had developed this test with an agency called BARDA. And part of that process was that we had to develop it in under clinical type rigor. And at the same time, we had to be able to test, do over a couple thousand tests a day. And so we had the robots in place, we had the expertise in place, and we thought, could we pivot that technology and switch it over to testing for the SARS-CoV-2 virus? And so somewhere around late February, we began that process. 7 of us from the lab would meet every day of the week, 7 days a week, trying to race against time to get that test running. That phone call that we had every day has blossomed into now over 70 people that meet on a regular basis to discuss how we're going to get that going. And by the beginning of April, we were running tests. So we went on what we called an offensive strategy against the virus. The idea was test as many people as possible, identify anyone who was positive, and if they were positive, get them separated from everybody else so they couldn't spread the virus to anyone. Once again, the whole goal here is to interrupt the transmission of the virus so it doesn't spread to people and it doesn't lead to people dying from the virus. So the more testing we can do, the less people will die from the virus. I do want to mention a little bit about what kinds of tests are available for those of you who don't always follow the science of all this. I'm going to start, let me just pull this up here. I'm going to start over here, the serology test, the antibody test. Serology tests are blood tests. These tests are designed really to determine if you had the virus in the past. You won't make antibodies against this virus for at least 10 to 14 days after you first get the virus. So you wouldn't expect the serology to be positive in someone who's immediately infected with the virus. But the blood test is useful for knowing where the virus has been, so that contact tracers and others can determine how the virus is traveling. Currently, the serology test will become useful for determining if people who've had the vaccine develop a strong response to the virus. I'm going to skip the at-home test and focus on these other 2 tests. One is called the Antigen Test. This is a test, it's done with a nose swab. It's typically done on the front part of the nose. And it's looking for a protein from the virus. The goal of the antigen test, it's called the rapid test because it gives a result in 15 minutes. And it is useful for determining if you're infected now, if you have symptoms. The antigen test is not particularly useful if you are asymptomatic. In fact, a recent study by our colleagues at the University of Arizona showed that in people who are asymptomatic, the antigen test misses about 70% of them. So it misses a lot. The test we use and that many people rely on as the gold standard test is this PCR test. It's the most accurate test by far. The test that we use tests down to 200 viral particles per sample. Keep in mind that most symptomatic people, or even most infected people, typically have 10,000 virus particles per sample. So it's a very sensitive test. And it once again is designed to determine if you have the virus right now, if you're infectious. In the version that we use, of course, we're doing it from saliva. So as I mentioned, we realized that we were well positioned to do clinical testing. We had a whole research institute with people who think creatively. We had robots in place. We had folks in place who could set up the logistics for collecting samples. The key was to think broadly and move fast. When we first started this operation, we talked to the various hospitals and clinics around the state to see what they were thinking. At that time, the number of nasal swabs that were available was around a couple thousand in the entire state of Arizona. So obviously not nearly enough for what we would need. And so we put someone in charge just for finding swabs. And the kits that we had to put the swabs into, we made ourselves because as a research lab, we knew how to make that sort of thing. And of course, we put people in place to set up database development, people to do software for analyzing the assays, people to run the clinical assays, automation engineers. Pretty much we had to come up with someone to handle every different element, and they all had to run in parallel as fast as they could to get this thing up and running as fast as we could get going. by April, we were running tests already for the public, or at least for, I would say, partners in essential workers in the community, like folks who run the power grid and others who had to work in close quarters. So the team always made their decisions based on saving lives. Now, of course, we realized early on that the nasal swabs, the nasopharyngeal swabs, were not going to cut it because it just took too long to collect those samples. It was too intensive for using the swabs. It involved a lot of PPE. And of course, it required clinically trained individuals to collect the samples. And we really wanted those clinically trained individuals taking care of sick patients, not standing in parking lots collecting samples. And that's pretty much when we pivoted to developing the saliva test. And we're proud to say that ASU was the first place in the USA to develop public testing for saliva. It became easy to collect. We collect a lot of samples at the same time. We could collect as many as 250 samples per hour at some of our testing sites. And Carrie, whom you just met, was one of the key individuals in setting up that testing at the State Farm Stadium, where we run a high throughput operation for collecting samples. Eventually, we ended up partnering with the state of Arizona. So the Arizona Department of Health Services got together with ASU and the Biodesign Institute, and they underwrote the testing that we do now broadly across the state. So at any given week, we collect over 100 sites across the state of Arizona from just about every county in the state, particularly looking to collect samples from underserved populations who can't get their tests done any other way. One of our commitments has always been to get these answers quickly. Even when the commercial labs were taking a week to 2 weeks to turn around their tests, ASU always had its answer out in under 48 hours. And most of the time, our answers are out around a day after we get the sample. So we're pretty quick at getting our answers done. And to date now, we've done well over 500,000 tests for the state of Arizona. We're one of the largest testers in the state. In fact, even when the Arizona Department of Health Services gets their tests done, they get them through us. So we're providing tests broadly around the community, and happy to say that all of the public testing we do is for free. So people who want to get tested for the virus can get it done for free at any of these locations around the state. So let's talk a little bit more about where the virus is today. Pretty daunting numbers. So this is what we call an epi curve, an epidemiological curve that shows the deaths that have occurred on the dates that the deaths occurred in the state of Arizona. Most of you will remember we had this big surge in the summer that really peaked at the beginning of July, and we lost a lot of lives there. And then, of course, this most recent surge, the one that we're experiencing just coming off of now, has been devastating for the state. As of now, more than 13,000 people in Arizona have lost their lives to COVID-19. To put that in perspective, that outpaces any other cause of death in the state of Arizona. It is worse than heart disease, worse than cancer, and unlike those diseases which primarily focus on an older population, more than a quarter of those deaths were people who were middle-aged and younger. Up to 24 children have died from this virus. So it affects a broad community. It is a daunting illness. And that doesn't even begin to address the people who will have long-term consequences from this virus. So we're up against a pretty nasty disease here, and we need to do everything we can to stop it. I do want to mention, because I know it's on people's minds and I get questions about this all the time, there are emerging variants of this virus. Anytime you grow a biological entity, any kind of organism, up to very, very high numbers, you're going to get a lot of genetic diversity. You're going to get mutations occurring when it gets replicated. And that's true of viruses as well. The fact that we have so much viral burden on the planet Earth right now means that there are going to be a lot of variations. And some of those variants are going to become better competitors than their predecessors. And that is what we're seeing right now. So the one that we're focusing a lot on right now is this B117 variant that comes from the United Kingdom. This particular variant is very good at transmission. It transmits much more readily, up to 70% more readily, than the viral variant that Arizona has had up until now. Now, ASU was the first place in the state of Arizona to identify at least 3 versions of this variant now in the state. We found that last week. That was reported in the news recently. Part of that was because the test that ASU uses to look for the virus has a unique pattern when the UK variant is present. We test for 3 genes at a time for the virus, and one of those genes will disappear if it's a UK variant. And so we noticed that pattern in early January, and Vel Murrigan, my co-leader for the clinical testing lab, identified that, and he immediately contacted Ephraim Lim, who is a genomic researcher in our institute. And Ephraim then did sequencing of those variants and identified them as the UK variant. And this sort of speaks to the unique characteristic of the Biodesign Institute, is that we have both researchers and people doing clinical testing all under the same roof. And so, Ephraim has his lab, 1 floor away from where the clinical testing lab is. And so Vel could hand the samples right off to him, and he could get that sequencing done almost immediately. And so that's how we found those 3 variants so quickly. So far, the other 2 variants listed here have not yet been detected in Arizona. The South African variant B1.3.351 and the Brazilian variant P.1 are a bit concerning, because unlike the UK variant, those 2 variants seem to be somewhat more resistant to the vaccines. Now, I will mention that even though they are more resistant to the vaccine, they are still susceptible to the vaccine, just not as much as the B117 variant is, or the variants that are currently in Arizona. So, but we are on the lookout for those variants, and we've been talking to the state now about by increasing the level of sequencing monitoring that we're doing at the Institute, and we will be doing a lot more sequencing now to monitor these variants. My own perspective here is that we're a little bit in a race right now between the vaccine and the variants, and we're hoping that we can get enough people vaccinated before the variants take over so that we can limit the spread of the virus. This is just to give you some sense of the Institute back in the days when we could walk around without masks on all the time. Hopefully you can see the video here. These are some of the robots that we use to run the assays. And this is sort of what the assay procedures look like when we run them. I don't know how this video is working. We'll have to find out later if it played well. I can always play it later. But
well.
Is okay?
Yep.
Okay. Yeah, I will say, kudos to ASU. We had to build a testing lab that was safe to operate in, and they built that lab for us in about 2.5 weeks. So the entire lab that you just saw was constructed in about 2.5 weeks. This just highlights some of the clinical testing that's being done at the stadiums. And I should mention that now the state vaccination sites, the stadium at State Farm, and now the Muni Stadium in Tempe, are being run by ASU teams. And so the ASU teams are also now delivering vaccines at the state sites. People can drive up 24 hours a day to get the vaccination at these sites. And this, the CDC recently stated that the stadium site that we run is the best drive-through COVID vaccine site in the country right now. So we're quite proud of the team and what they've been able to accomplish. I do want to briefly mention a little bit about the vaccines. I know some of you may have questions about that, and we'll get to that shortly. The current 2 vaccines that are authorized for emergency use in the USA are both mRNA vaccines. I know some people have commented on how quickly they were successful at testing these vaccines. I will point out that people have been developing this methodology for years. So, after many years of research, they are an overnight success. They were not invented this year by any means. The vaccine uses a small piece of genetic material that encodes a piece of the virus. This material does not last in your body. Once it's used to make the piece of the virus, it disappears, it gets digested. But it makes enough of the virus that it alerts the immune system in our bodies that what the virus looks like. so that our immune system is poised to attack that virus if it ever encounters it through an infection later. This is much like sharing a mugshot of a criminal to police offices and post offices around a community to say, be on the lookout for this bad guy. If you see it around, arrest it immediately. And that's really what this virus does. So we inject people's arms with this. I will point out that this vaccine is not fully effective after the first dose. You really need to get both doses before the vaccine is functional. The first dose kind of primes the system and it alerts it to what the virus looks like. The second vaccine dose is the one that really induces enough of a response to be protective against the virus. We have seen documented cases of people who thought they were immunized after one dose, who then got infected because they lowered their guard and they went out partying and they didn't wear masks and they got infected. So you really need both doses to be protected. I think that that's most of what I have to say. I'm going to stop here. I know probably people will have questions, and the goal of this is to have a conversation. So I'm going to hit escape here and stop my sharing. And we can go to the Hollywood Squares thing, and people can ask questions.
Yeah, so Josh, we did have a couple of questions come in through the chat. So if you would like to address first, Lisa and Chris Bain sent over a couple really good questions regarding vaccination. They say, since the present vaccines target the spike protein, surely it will be cell adhesion and infection that should be blocked. If so, why is there such concern about the continued susceptibility and transmission among vaccinated individuals or survivors?
Okay, very sophisticated question, very good question. Let me, of course, this crowd would ask smart questions like that. No surprise there. So let me comment on that. So yes, the two vaccines that are currently approved in the U.S. Both address a part of the spike protein. This is the protein that allows the virus to invade our cells. And in fact, the idea is to induce the body to make antibodies that will prevent the virus from infecting cells. The clinical trials that were done, the end point that they looked at was severe disease. They did not look at infection per se. They specifically looked at, do people get severely ill? And so what we can say for sure is that both of the vaccines that are out there right now are around 95% effective at preventing severe disease. The person who asked the question is asking, well, does it prevent infection at all? Because it's designed to block the invasion of the virus into cells. Truth be told, it does limit infection, but it does not prevent it entirely. It's about a threefold benefit in terms of preventing infection. People who get vaccinated can still get infected by this virus. They just don't get severe disease, so they don't get as infected. So what we are advising people right now that they should assume that even though they've been vaccinated, they might still get infected and they might still be viral carriers. Now, once we achieve herd immunity and the virus can't move around so easily because so many people are immune that the virus can't keep infecting people, then the level of virus in the community will drop significantly and we don't have to worry about that so much. But right now, when the virus is rampant, and it still is rampant, today the viral level, the number of the cases level dropped just below the peak of last summer. So while it is now lower than the peak of last summer, it's still up near the peak of last summer. So it's not by any means gone. We would recommend that you still, when you're in public, wear a mask, even if you've been vaccinated. I have been vaccinated and I still wear a mask in public. And then that you still, to some extent, limit your circles, keep it close to the immediate family for now, even if you've been vaccinated, until we can get more of the community vaccinated.
Thank you for that. We have a flood of questions coming in. So next up, could you address some of the kind of skeptical nature around the PCR test Jan Smith says, how can any numbers be trusted when the inventor of the PCR test said it was never designed for public diagnosis? Should use the term vaccine when these treatments don't meet legal definition of a vaccine. Can you speak to this at all?
Okay, well, so I'm not sure why there's a concern about using the qPCR test for doing diagnosis. I mean, there have been a number of qualified, certified diagnostic methods that use qPCR for testing. So qPCR testing is a clinical test and can be used for clinical testing. It basically is one method for measuring the amount of nucleic acid in a sample. And the test that's being used currently for detecting the SARS-CoV-2 virus can detect down to around 200 viral particles per sample, when typically clinical samples have around anywhere between 10,000 and above particles. So it's quite sensitive at detecting it. So I think, I mean, when the method PCR was first invented, which was many years ago now, that wasn't what was on the mind of the inventor, but it has been adapted and tested and is quite reliable. The vaccines that we're using behave exactly as vaccines should. They clearly induce an immune response in people. You can detect that by looking after the fact and show that they have produced what are called neutralizing antibodies. These are antibodies that block the virus, and you can show that in people who've been vaccinated. And so they are doing exactly what we want them to do. Of the people in the clinical trials, and when I say clinical trials, I mean roughly 30,000 people were tested in each of the two clinical trials for the two different RNA vaccines. Among those 30,000 people, of the severe cases, none of them had received vaccine. Only people who had gotten those severe cases had received placebo. So the vaccine clearly works at preventing severe disease.
Okay, thank you. Can you also speak to some of what you mentioned about the variants? Are the variants of this airborne, or do they spread in a different way? And then also, how does the variant nomenclature work?
Right. So yes, the variants are still SARS-CoV-2 coronaviruses, and they still spread by an airborne route. I am not aware of clear evidence as to why particularly the B117 variant is so communicable. I don't know exactly. I think some people believe that it latches on better to cells, but I don't really know the details for, or if it's even known why that variant is so transmissible. The evidence that it's more transmissible is that in a population of individuals, particularly in the UK, that initially had many different variants at one time, all of a sudden they noticed that this particular variant predominated, which means it outcompeted all the other viruses to become the most prominent strain. And that's the evidence that suggested that it was more transmissible. The nomenclature really comes back to these genomic wizards like Ephraim Lim, one of the faculty members in our institute. And what they do is they sequence the entire virus. It's around 30,000 bases. They sequence all of the DNA in that virus. And then they use sophisticated software to line up viruses against other viruses, and they look for changes. It turns out that the B117 strain has about 17 different changes compared to other variants, or the one that we're used to seeing here in the US. And one of them happens to be in this S gene, but many of them are in other parts of the virus. And then based on how they relate to other viruses, they give them numbers like B117 or B1351 or so on and so forth. And it's just a way for them to keep track of which ones they are. Some of these different strains are related to others. They have some similarities and other differences.
Thank you. Okay, so this next one's kind of two-part, two questions coming together. For one, can you discuss the future importance of boosters? Is this going to become, part of our everyday life for years to come? And then spinning off of that, someone said they encountered an anti-vaxxer today. Do you have any advice on how to combat that?
All right, well, let's start with, the booster idea. So It's worth reminding everyone, we in the context of the flu virus, another virus, that we are often recommending that people get the flu vaccine every year. And that's because unlike this virus, the flu virus is very sophisticated in terms of changing its disguise, its molecular disguise. The flu virus every year changes what it looks like, and different variants show up in our community. And so every year, people who study the flu do their best at predicting which disguises the flu is going to be wearing this year. And they produce a vaccine against multiple different versions, typically 3, so that people are prepared for the most common likely flu variants out there. It turns out that this virus isn't so sophisticated about changing its appearance. It doesn't mutate nearly as fast as the flu virus does. And it doesn't change its look that much. Now, right now, we're faced with a big problem because there are just so many copies of this virus out there that even though it doesn't mutate frequently, there's just so many out there that it is still mutating. Once we get that under control, I don't think we're going to have to worry as much about boosters for this virus as we did for the flu virus. Still, there are multiple variants out there, and we may, until we get it under control, have to get some boosters, particularly boosters that address any variants that emerge that are resistant to vaccine. If we can get under control, we can limit the spread a little bit. It's hard to know what to say to an anti-vaxxer. To those of you who are scientists, I can tell you this. It's because of the anti-vaxxers, particularly those that were worried about autism. And let me just briefly mention that the one study that suggested that there was a link between vaccines and autism was withdrawn because of fraudulent data. That particular study was discredited, and all of the authors of that study have withdrawn their names from it. So there are no data to suggest there's a link to autism. But because of that crowd, large studies were done on the long-term consequences of viruses. And when I say large studies, I mean millions of people were studied. And in all of those studies, there has not been any evidence that there are long-term consequences of vaccines. So let me say that once again. There are no long-term consequences of vaccines. If you're going to have a consequence of a vaccine, it's going to be almost immediate, and most likely it will be due to either a strong allergic reaction to something in the vaccine, or a chemical sensitivity to the vaccine, and it will become apparent almost momentarily. And those of you who've had the vaccine already know that they want to watch you for 15 minutes because they're looking for that response. But beyond about 6 weeks after you've had the vaccine, there are no consequences of vaccines. Now, will that work, telling that to someone who's an anti-vaxxer? I don't know. Psychologists will tell you that those people are not interested in hearing about data. It's more of a personal thing for them. And so I'm not the best person to ask how to counsel them. I will tell you this: when I've encountered people who are nervous about the vaccine, the thing that has worked the best for me is to tell them, look, either I'm going to get it or I've already had it. And if I tell them that I personally put my arm where my mouth is, it does seem to have some impact. I've had both doses now, and I did have a sore arm for about 24 hours, but that's about the extent of my side effects. And I'm here talking to you now. I've had no other consequences from the vaccine. And I can tell you without a doubt, whatever soreness I had in my arm, I would much rather have that than the virus, because the virus is far worse than the vaccine is.
Thanks for that. Okay, we've got another good one coming in. This is over my head, but are the adjuvants in this vaccine similar to other vaccines in current use, or does the mRNA vaccine require different adjuvants? I'm not sure if
that's a good question. That's a very, again, typical of a biodesign crowd. You guys are very sophisticated here. So those of you who know about vaccine technology know that in the early days of vaccine development, people would add something called an adjuvant, and it's a chemical that's added to the vaccine to kind of induce inflammation at the site of the vaccine. And by inducing inflammation, it brought more of the immune cells local to the vaccine and caused a better immune response. And that's what the adjuvants were intended to do. In the case of the 2 mRNA vaccines, we don't need to use adjuvants. This technology is so sophisticated, it does not require them. In fact, if you look at the ingredients in the mRNA vaccines, they are actually quite simple. So, for example, there were people who would develop allergic reactions to previous vaccines because products from eggs were used in them and other molecules that caused an an allergic response. You won't see that here because we don't need them here. These vaccines are actually quite simple in their formulation, and therefore you don't really need adjuvants now. And now that may not be true for some of the vaccines that are coming down the road. I know many of you are watching those. The J&J vaccine is about to come available, and there are a few others down the road that do use proteins, but I'm not sure if they even use adjuvants for those.
Great, thank you. So I've got a fun one for you from Richard Haisner. He says, what research in this area are you currently not doing but wish you were?
Well, we're just getting ready to start looking at the immune response that people undergo as a result of vaccination. Our lab is currently developing a serological test that will look at not only the immune response to the spike protein, which is what the vaccine is designed to induce, but also to other proteins, both from the other proteins in the virus, as well as other proteins from other related coronaviruses. One question we'd all like to know is, what is it that makes some people have such a profoundly terrible response to this virus and other people not nearly so bad? What is it that makes some people so susceptible to the terrible consequences? I think another important area that we'd love to do research on, and some of us are working on getting there, is there's a lot of emerging evidence now that some people who've had an illness, not necessarily even people who went to the hospital with COVID-19, but who are now having long-term consequences of the virus. Headaches, something called brain fog, some cardiac symptoms or cardiac damage. And these people are not getting better quickly. They continue to have symptoms months and months after their infection. And I think we really need to understand what's causing that and what can be done to prevent it. And I think that's an important area that we have to start thinking about, because after this pandemic, there will be a lot of those people.
Thank you. Okay, so another question from our audience here. There have been rumors that say you shouldn't take pain meds before getting your vaccination, but it's okay afterwards. Are the pain receptors somehow used to bring in the mRNAs?
I'm not aware of any significant evidence to suggest that if you want to take an ibuprofen, either before or after you get vaccinated, you shouldn't. I will tell you this, having had both doses of the vaccine, you don't feel the injection at all. It's really nothing. And the soreness in your arm does not start for about 10 to 12 hours. So there's really no need to take pain medicines before the vaccine. Maybe 10 to 12 hours after you get it, some people will feel sore and maybe an ibuprofen would help them. But I'm not aware that that's a contraindication at this point.
Awesome. Kind of along those lines. So talking about vaccines again, is there a current effort to ascertain T-cell immunity following vaccination?
So the 2 vaccines that are currently out there, the 2 mRNA vaccines, appear to induce multiple types of immunity in our bodies. So again, that's a bit of a sophisticated question, but there are 2 major branches to what's called the adaptive immune system, and that's what we're talking about here. The adaptive immune system is the part of our immune system that trains upon and learns about our enemies. It learns about the pathogens that it infects us. So it recognizes the flu, or it recognizes the chicken pox or whatever. And one branch of that system is the antibodies. Those are the little free-floating molecules that bind to the invader and set up a signal that tells the body, hey, I found him. Here's the perp. Come and get him. The other part of the immune system is the cell-based immune system. It's often called the T cells. And these are the cells that come by and either engulf the bad guy or inject the bad guy with something that kills it, or in one way or another kills the invader. And it turns out these mRNA vaccines induce both branches. So they induce the antibodies, but they also induce the T cells. Now, I don't know enough about the new J&J vaccine. It's a viral vaccine that it's a DNA virus that injects, again, a piece of the gene that induces an immune response. I suspect it will also induce the T cell immunity, but I don't know too much about how much that's been studied yet.
Thank you, Josh. Okay, I think we have one more, unless there are any more flowing in, but there's been a lot of talk about artificial intelligence. I know, so much of what you do is on the cutting edge. Is AI playing a role in this research? If not, will it in the future?
So the work that's been done so far in terms of developing the vaccines did not necessarily involve a lot of artificial intelligence, although one key element that made the whole vaccine field successful here was some work done early on by some investigators who figured out ways to make specific mutations in the spike gene that stabilized the shape of the protein so that it would take the conformation of the natural virus. Without those mutations, I don't think the vaccines would have been successful. So that work was pretty crucial. I don't know how much AI was used in that, but certainly going forward, I think AI will be critical. I think in everything we do now, we invoke this, what's called machine learning technology or deep learning technology. It's a way of kind of statistically better understanding how different things happen. And I suspect that particularly for example, I mentioned earlier this area of research around what are called long haulers, people who get this virus and have long-term symptoms. I think AI will become very important in understanding what that is about.
Excellent. Okay, I think just one or two more here. Is there anything that we need at Biodesign to accelerate our research efforts for COVID or anything else?
Well, of course, our institute can always use support. Research is not inexpensive. It involves, we need resources. One thing we need a lot of, I think, moving forward are people willing to volunteer for clinical research. The only way we're going to understand how this virus acts on the human body is by measuring what happened to people who either had the virus or who got vaccine for the virus or who didn't get the virus despite exposure. So, we're going to need people who are willing to volunteer to participate in clinical research on this. But any kind of support was always welcome for sure.
And then kind of spinning off of that, now that Biodesign has this clinical testing lab and all this experience in this space, how do you see the future of Biodesign changing? What's it going to look like?
Well, it has been revolutionary for us, the clinical testing lab. I mean, if you had, for years and years, since I've been at Biodesign, we've been wondering about whether we could set up what's called a CLIA lab, a lab that could do clinical research and provide results back to people. Keeping in mind that ASU is a university that does not have a medical school. And so traditionally, ASU never had direct interaction with patients before. And that's been a challenge for us because in order for us to do clinical research, we always had to partner with, one of our good partners, which is the Mayo Clinic, or with Banner, or with Dignity Health, or Brazos, or some of the other clinical providers in the Valley. And so it's been true that it's been good that we could interact with many of them, but we were always one step removed from patients. And one of the things that the clinical research lab the clinical testing lab has done for us is that it put us in direct contact with patients. And at this point now, we've dealt with hundreds of thousands of them. That's how busy our clinical testing lab has been. And I think that has changed the way we look at things now. in a way that I think will be permanent for us, because now we have a direct and personal relationship with people who need help from us, and we can turn to them and say, you know, we need volunteers to help us with our research. And already we're getting people who are signing up, and we are already starting to think about other types of clinical tests that we could develop. For example, we realize, and it's not so much this year, but in future years, there will be a challenge in terms of differentiating when people come to us with clinical symptoms of a respiratory infection. Do they have COVID-19 or do they have the flu? Because they look the same. And so we're now in the process of developing a test that will simultaneously ask, is it COVID-19 or is it the flu? And so being able to develop tests that can distinguish among multiple respiratory infections that might simultaneously infect people, I think will be very valuable. And so we're looking forward to other clinical tests we might be able to develop at Biodesign.
Perfect. And then, you know, speaking of our clinical lab, how can people sign up for COVID or other research studies at ASU? What do they need to
Well,
do?
thank you for that question. I'm suspecting that some of our friends planted it for me. When you sign up to get a COVID-19 test from us, one of the early questions you get asked is, are you willing to participate in clinical research? And we would, of course, welcome it if you would say yes, and let us contact you for possible clinical research. If you go to any of our testing sites, there's a VR code. You can flash your phone at the VR code and it will automatically bring you to a site where you can very quickly click and link to being willing to volunteer. And I suspect on our website somewhere there's a place where people can sign up. And although I don't actually know if we have that, so I'm kind of looking at Carrie right now and
Yes,
saying,
we do. We do indeed.
yeah. So yeah, we would always welcome people willing to volunteer to be participants in clinical research. It's, you know, many of you who know me know that my background is I'm a cancer doctor. I don't usually do viral studies. I usually do cancer diagnosis. And one of the things that is a foundation for me is that the only way we ever get better at treating our patients is by doing clinical research. That's how we improve clinical care. The old adage, and I know some of you are physicians, the one thing that we always were taught in medical school is that half of what they teach you in medical school is wrong. The question is which half? And you know, we have to always learn from our mistakes. We never get better if we don't look at what we do, take that data down, interpret it, and learn how to get better about it. That's what we have to do.
Excellent, thank you. I think there's one or two more here.
I thought I've exhausted you guys by now.
That's a good question.
You guys keep coming up with new questions. That's great.
Going back to, you know, the science side of it, someone asked, why, in your opinion, is there such slow progress in looking for genetic markers of disease susceptibility?
Okay, so this is a more broad question now. I think we're kind of talking more broadly about disease susceptibility. So, you know, I mean, the challenge that we're up against is it's a numbers challenge. You know, human beings are so varied from one to the other. And although fundamentally our overall genomes are very similar, there's enough genetic diversity amongst us and enough just life diversity amongst us in terms of what we've been exposed to, in terms of our habits, in terms of all the different things we do, that the things that can lead to susceptibility can sometimes be subtle. And you really need to look at very large numbers of people to be able to determine if something is having a profound effect. So some things we're very good at detecting. For example, smoking. Smoking is a huge risk factor for cancer. Smoking by itself is a greater risk factor for cancer than all of the other risk factors combined. So that's an easy one to spot, right? But then there are other things like diet and fat in the diet, and they are much, much more subtle, and the effect that they have is smaller. And so you really need large studies to be able to tease that apart. The more research we do, the more we'll pick those things apart, but it's not simple.
Great questions. All right, I think they're still coming in a little bit here. So we got one from Robert. Is there a possibility that the upcoming vaccines based on adenoviruses may limit future applications of these viruses as vectors?
It's a good question, and also a very sophisticated question. So we have a very smart crowd here tonight. Yeah, so the J&J vaccine is what's called an adenovirus. It's a virus. Let me first say that it's a crippled virus. It's a virus that once it infects your cells can never replicate or make more of itself. So it's designed to deliver a payload to our cells, but never again be useful again. And what it delivers to our cells is a piece of the SARS-CoV-2 virus that will allow, once again, kind of a mugshot, if you will, of what the virus looks like, so our immune system can prepare itself. The concern that this Robert is asking is, once our body has seen that adenovirus, will it then prevent future adenovirus vaccines from working? I don't know enough about the design of the adenovirus, but I suspect that the people who designed it have been thoughtful about that, and they've tried to limit the number of immunogenic elements of the virus itself, so that when we deliver the payload to the cells, we're not inducing an immune response against the virus. Now, that doesn't mean it can't happen, but I think they've tried to limit that.
Great. I know we're getting close to time here, but there's still a few more questions. We got one here. Statistically speaking, shouldn't mutations in a virus lead to less virulence, less danger, less problems?
Well, yeah, yes and no. So, viruses have been with human beings, or frankly, with biology since the beginning of biology. These, viruses, if you think about it, it's like a little piece of paper in an office that says, "Copy me." And some unsuspecting worker picks it up and says, "Okay, I'll make a copy." And they make a copy. And what viruses have done over the years, and when I say years, I mean the millennia, I mean the millions of years that viruses have been with us, is they've come up with even more and more sophisticated ways of convincing office workers to make copies of themselves. Now, the best viruses are the ones that cause the least disease, right? Because the less disease they cause, the more willing people in the office will make copies of them. The problem with this particular virus is that it's brand new to human beings. I mean, it first jumped over to our species November of last year, of 2019, basically. That's when it first entered the human race. And so it is not very sophisticated. Over time, with lots of mutation, it will adapt itself to make us less sick. But unfortunately, right now, the virus is most concerned with spreading itself as far as it can. That's the biological imperative, right? That's what evolution is designed to do, is to make more copies of itself. And right now, the viruses that are out there, the vast majority of the mutations are probably deleterious to the virus. They probably prevent its spread. But with so many copies around, with so many billions and trillions of copies of this virus on our planet right now, sooner or later, one of them is destined to be better at transmission than the others. And if it is destined to be better at transmission, then it will start to overtake the local population and become the most dominant strain. And that's exactly what happened with the B117 strain in the UK, is that this particular virus happened to be a little bit better at transmission. And because it was traveling around in that community, sooner or later, it became dominant there.
Awesome. I think we have just one more question, kind of a good one to wrap up on here. So we've been reading things about vitamin K, D, and C to help fight COVID-19. What is your opinion on that? And then, can you wrap up by kind of just speaking about what can we all do from here on out besides everything we've already talked about?
Right. I don't know too much about vitamin K. Vitamin K is involved in clotting, and I'm not sure that would necessarily play a role here so much with the virus, except to say that there have been some clotting abnormalities with this virus, but vitamin K would only make you clot more. Some people have talked about vitamin D. Vitamin D is a vitamin that does support the immune system. So I could imagine that taking vitamin D wouldn't hurt. And frankly, many of us in Western culture are vitamin D deficient. So even though Arizona has a lot of sunshine, most people don't spend enough time in the sun and probably shouldn't. So vitamin D wouldn't be bad. Although I don't know of any specific evidence to show that it prevents consequences of this virus. The best advice I can give people right now, and you're going to hate me for it because it's what you've heard a gazillion times, is we really don't want to spread this virus. And so we have to continue keeping our personal circles small, stay, keep the people that you're with, that you live with around you, that's fine. But if you're visiting people that you don't see all the time, you're better off wearing masks. You're better off doing it outdoors where the air is better. And we're thankfully in Arizona, we can all smell the spring now. It's all in the air right now. The weather's getting warmer. It's going to continue to get better. So do what you want to do outdoors more if you can. Wear masks when you're around people that you're not around all the time. Wash your hands. All those things right now will prevent the spread. And of course, as soon as you can do it, get vaccinated. We want to get everybody vaccinated. The vaccine is really the light at the end of the tunnel for us. It's going to get us out of this. So get it as soon as you can.
Great. Well, with that, I wish I had like an applause audio track that we could that we could cheer for you, Josh. It's great. I see some high fives and some hands in the air. We want to thank you, Josh, for your preparations and really for all that you do. It's an incredible team at the Biodesign Institute and proud to be a part of it. Thank you so much. You all did such a phenomenal job. Putting the questions in the chat. Oh, great. Thank you for that. Thank you for the clapping. Yay. Oh, man, what an incredible crowd here. We just so appreciate all of you. And I hope you've had a chance to notice the chat. There have been a lot of interesting little updates and hyperlinks for you about how to subscribe. We have an incredible Biodesign newsletter. And if you'd like to learn more, we invite you to share that's in the chat right now. Also, there was a question about how to join our research studies. We've got a fantastic website that is available. And let's see. My screen has gone a little goofy here for the moment. There we go. But very, very grateful to all of you. I'm going to share my screen here as we conclude. Here we were planning to do an open Q&A, but we were flooded with your fantastic questions. So really appreciate that. But I just want to let you know that this talk is going to be available on our website on the Sip of Science tab at our Biodesign at asu.edu. And I also just want to share, don't forget, we have some incredible other experts that are going to the first Tuesday in March and the first Tuesday in April. We have some great additional things to share with you.
At least one of them is here on the call. I see him.
Yes, that's right. We have Dr. Tim Long here. So we've got some more experts. So here we've got 1 month from today, we've got Grant McFadden, who's going to give us a fantastic talk on viral overload. Do viruses impact cancer? And for next time, we are going to have not a pre-recorded, but we're going to have a real-time virtual demonstration from Ruth from Q3L Cuisine. So we're going to have a fantastic menu, and that's going to be some mushroom deluxe turnovers. So really looking forward to her doing that for us in real time. And then Tim Long. Tim, who's here on the call. We are so thrilled. So he is a new leader here at the Biodesign Institute. And drowning in plastic, what are the alternatives to keep people and, importantly, our planet safe? So really, really looking forward to that event. And finally, as we conclude, so many of you are fantastic supporters here of us at the Biodesign Institute, but many of you are our Pioneer Circle members, and we appreciate the annual donation of 1,000, but many of you have given so much more, plus your time, your energy, your advocacy, your ambassador Chip, for what we do at Biodesign, and can't thank you enough for all of that here. So we hope that if that's something that you're interested in considering, please, let me know, or you can reach out. We've got Elizabeth Munkle, Eric Spicer. We've got others here who, many of you know well. But with that, I just want to thank you for joining us. And we hope to see you all again soon. And I just want to say thank you and good night.
Good night, everybody.
Bye, all. Take care.
Bye.
Thanks for joining us. See you next month. Bye, all. Thank you. Have a good night. Thanks, all. Oh, it's great to see your faces. I'm missing so many of you. Oh, it's great to see you.
Thanks so much, Carrie.
Thank you. Thanks, all. Good night.